Hello and welcome everyone to our single-cell transcriptomic workshop, a live mentor-guided training designed to take you from the raw data to the real biological insights. Let's take a look at what you learn each day. The day one is about introduction to single-cell data. On day one, we will set the foundation and you will learn about how to access and explore the public cell datasets. You will understand about the data formats and how to prepare your data for further analysis. Get hands-on quality control, normalization and pre-processing using popular tools and open source platform. The day two is going to be about dimensionality reduction and clustering, which tells us about the data analysis workflow. On day two, we are going to make sense of the complex data. We are going to dive into the dimensionality reduction using PCA, DSNE, and UMAP, then perform clustering to identify distinct cell populations. Here you will discover hidden structures and relationships between the cell like never before. The day three is going to be all about cell type annotation and functional analysis. Here you will learn how to annotate the cell types using known markers, explore differential expression between clusters and identify key genes that define the cell identity. Then you will perform the pathway enrichment analysis, visualize your findings, and you will learn how to communicate your results clearly. Whether you are a student, researcher, clinician, or a bioinformatician, this workshop is your gateway to master single-cell data analysis. Live sessions, expert mentoring, and real-world data, all from the comfort of your screen. So enroll now and take the next step in your omics journey.